This is on. Okay. Great. Well, welcome back, everybody, after the break. And we're on to our second session today. We're about 15 minutes behind schedule, so we'll just try and make up a little bit of time. And we may even keep questions to the panel, but discussion time, but we'll see how we go. So our session 2 today is on gaps in treatment. Can everyone hear me in the back? Yep, perfect. Okay, we're going to use the roaming mic now for presentations, just so it's a little bit easier for people. So we've got 3 speakers today. First, we've got Ben Rodgers, fitting therapy of AMR infection to the patient and the infection. We've got Sam Forster at AMR and the human gut microbiome. And then we have our last speaker, Philip Andrews, who will be talking about lessons from antiquity, metal-based antimicrobials. So if I can find our bios here, which I have a feeling are at the front of this document. Yes, at the back. Thanks, Nanette. After our bios. So Ben is an ID physician at Monash Health, and he asked me to keep this nice and short, so I will. But his research is on the epidemiology and management of multi-resistant gram-negative infections in Australasia. So Ben, over to you now.
Thanks so much for the introduction, and look, it's really fantastic to be speaking today. I'm really conscious that this is quite a mixed audience, some clinicians, some scientists, lots of other people. So I'll try and explain a bit around my talk, which is somewhat clinical in nature, but hopefully will give you an understanding of some of our unmet need in this space. So the other hat I wear in research is also in this program called Hospital in the Home, which is what a bit of my talk's going to be about. And I'll explain that as we go. So I'm happy that, no relevant declarations first. I'm happy that Nenad gave us that diagram of new antimicrobials, because this is really going to be focusing on some of the challenges with new antimicrobials. And when I talk about new, I mean ones that we actually have now to use. There's an amazing amount of stuff coming on. Not enough, but a great amount. But today I'm not going to talk about that. That would take another whole full-day symposium to cover. So, in fact, between 2017 and 2022, we've had 9 new antimicrobials kind of registered by the European and the North American registration bodies. And really, that was mostly up until 2020, because after that, they obviously changed tack and started registering antivirals as fast as they could. We've had a number of agents for gram-negative infection. We've had some new beta-lactamase inhibitor combinations. These have mostly been focused on what we call class A beta-lactamases, KPCs, if you've heard of them. But there are a number of other gram-negative resistance mechanisms they don't cover very well. These are kind of derivatives of ceftazidime avibactam, which was the kind of first in class of these. We've had this new, somewhat novel agent, a siderophore cephalosporin called cefidoracol, and this is probably as close as we've got to kind of, a card-carrying superbug-fighting antibiotic. And then a new aminoglycoside called plasmamycin. And look, really, that just sums up the challenge that, as I've mentioned, of, producing and selling a new antibiotic. Achaogen, the company that made that and brought that to market at a cost of about $2 billion, actually went bankrupt within weeks of it coming onto the market. And just showing some of the real issues with the economics of this at the moment. For gram-positive infection, we've actually had a lot more diverse range of antibiotics, and I'm going to talk about this in the next slide. We've had some new classes of antibiotics. We've had some kind of upgrades on old classes of antibiotics. But of note, there's IV formulations, there's oral formulations, and there's quite a bit of diversity in what's come along. And I think one of the first things to ask about this is, why is there such a difference between the two? They're all bacterial infections. They all infect humans. But for the gram-negatives, we've really just seen the advent of new intravenous therapies only. And that line there actually comes from one of the many reviews of new antibiotics you can find online, which really explains why the gram-negative antibiotics are really parenteral therapies designed to be poured into septic and sick patients in hospital, because, really that's where the pressing unmet need has been in this space. It's been in acute sepsis. We know thousands of patients die every day. We know, in neonatal sepsis, there's huge rates of antimicrobial resistance in the developing world. We really need these antibiotics for that space. And also pharmacologically, the focus has been on beta-lactam therapy, which is, for various reasons, quite a bit harder to give orally than other antimicrobial classes. And I've just put there, one of the antibiotics that unfortunately didn't make it onto that list of new antibiotics, called tebipenem, which was kind of a hope in oral therapy for gram-negative AMR infection. And in fact, some of you might have seen the NEDGEM paper on the kind of the clinical trial of this last year, which was positive. But when it got to registration, in fact, the FDA, for some pretty reasonable reasons, wasn't happy to register it on the data that was provided, really just highlighting the challenge of these things. Whereas for the gram-positives, and these are things like, skin and soft tissue infections, pneumonias and things like that, often caused by, staphylococci, streptococci. There are a lot more varied clinical syndromes that these agents have been developed for. That SSTI is just skin and soft tissue infection. I think there's another S in it, actually. And the classes of drugs that have been used are a lot more varied as well, which has really allowed, for agents that are easier to give orally and deliver in ways other than, through an intravenous cannula in a hospital. So I guess if you kind of take all of this and mix it all together, what I'm saying is that, when we look at, new agents and new therapies, as well as the bacteria and the resistance mechanisms, which is something that we tend to get very focused on as kind of scientists and sometimes as clinicians, we also have to think about the syndrome and the setting where we actually need to give those new agents and make sure that we can, fit all of those things to the therapy. And, Katrina's chairing this session, and, the STI is a great example of a space, that's kind of outside of at least us hospital clinicians' normal thinking for where we try and treat infections. And, I guess we have the community treatment of resistance, we have the hospital treatment of resistance. And what I'm going to talk about a little bit is this crossover space called hospital in the home, where I work, which is really about, trying to bridge that gap between the two and give some of this complicated treatment for antimicrobial resistant infections in the community. And I've just put a few emoticons here really to summarize where I feel like we're up to in this. And, in the gram-positives, either skin and soft tissue infections and things like that, we actually, have pretty good therapies, that work from hospital in that intermediate space, and also oral therapies that we can use in the community. Whereas for the gram-negative infections, these are our ESBLs, but our kind of more powerful mechanisms, our carbapenem resistance mechanisms and other things, we do have a number of emerging options in the hospital space. But when we kind of get into that intermediate hospital in the home space, and also into that really community space, people walking around well in the community who shouldn't need to come into hospital for treatment of, for instance, their urinary tract infection, we don't have new agents. And, this arrow is really just to highlight that, at times we actually have to pull people from the community into hospital just to treat what's an otherwise simple infection. So I've mentioned this hospital in the home or outpatient antimicrobial therapy space that I work in. And, this is really an emerging and important space in kind of antimicrobial therapy and healthcare in general, but also in the treatment of AMR infection. I'll kind of use these terms interchangeably. In Australia, we call it HIF, but in the rest of the world, they call it OPAT. So, you'll see both of those terms in the literature. And really, to explain for those of you who aren't in the clinical space, without being too technical, it's just giving patients intravenous antibiotics at home. And really, we use this when a patient needs an extended course of intravenous antibiotics. Sometimes this is because of a particularly complicated infection, a brain infection or a heart infection where we need to give a long course. But I guess the unmet need for us is really around infections where there aren't oral options, where there isn't a good tablet option or something that we can give easily, and we need to give intravenous therapy to treat the infection. Antibiotics, if you can't give, one squirt once a day, we only go to see the patient once a day, then what we do is we tend to hook it up to something like this, which is an elastomeric infuser. It's a plastic bottle with a balloon full of antibiotic suspension, and over the day, the antibiotic is just slowly infused into the patient through an intravenous drip, along this tubing. And once a day, a nurse will come to the house or the patient will come to us and we'll just swap this for a new bottle and reload it. And really, you don't have to be a rocket scientist to work out that people love this. This is a really good thing. There's a lot of data really to show that patients prefer to be at home rather than in hospital. We avoid all those complications of hospitalisation by treating people at home. The clots in the legs, the C. diff diarrhoea, the acquisition of new antimicrobial resistance, all of these things. And hospitals love it. There's a great saving of beds. We can use beds for sicker patients, more complicated patients. It probably works out about 60% cheaper than having somebody in hospital treating them. And just a real life example, this is my cousin, picture used with permission, who was on our program, just showing his antibiotic infuser in his left hand as he went for a ride on his bike. Probably not an approved activity. And this is not kind of a flash in the pan thing now. If you look Australia wide at treating patients with hospital level care in the community, and this figure includes antibiotic and non-antibiotic treatments, about 1 in 25 of all admissions to hospital involve this kind of care. Over 500,000 bed days a year. As I said, the safety and the economics are very well established, and we're really seeing this increasing, a proportional utilisation of this kind of intermediate space between the hospital and the home for treatment. This is just a figure that shows that the blue line is hospital in the home, and the green and the red lines are other activity measures, and what you can really see is they're converging. And for some AMR infections, in fact, we know that HIF is very heavily utilised. This is work from Ollie Z, one of our previous registrars, who's gone on to a lot bigger and better things. And we really looked at, what I guess is a problematic but relatively low-level resistance of ESBL E. coli. So E. coli that are resistant to most of our common, oral antibiotics. That can be treated actually with a once daily dose of ertapenem, which is a carbapenem antibiotic. And we in fact had in our health service, which is a big health service, 108 patients coming in over 2 years with this infection in their bloodstream, requiring kind of over 1,100 days of antibiotic therapy. So this is a really big volume of patients and a lot of beds. But in fact, with our hospital in the home program, we're actually able to give about 40% of this therapy. So, over a third of this therapy outside of the hospital walls. So this is a very effective, I guess, way to use our resources in this setting. But we really do have a big challenge, which is that, for our more difficult gram-negative infections, our more challenging kind of superbug infections, our carbapenem-resistant infections, even some of our carbapenem-susceptible infections, we just can't use hospital in the home at the moment. And really, without overstating it, the simple issue is that a lot of the new antibiotics that are coming onto the market, can't fit our programs. So, to be able to use them, we need to be able to give them once a day as a single squirt, which few antibiotics actually can do that because of the half-life of them is a bit short, or they need to be stable enough that we can fill them up in one of these infusers and leave them with the patient for the whole day. And the challenge is that most of our new antibiotics, especially in the gram-negative space, are not heat stable enough for this. When we fill them up and put them in an infuser, they start to break down over the day. And by the end of the day, really, the patient is not getting enough antibiotic through the infuser to treat them. And one of the reasons they break down is, when you give a patient an infuser, they put it in their pocket and it heats up. And, so when we test them, we're really looking to see that there's, less than 5% breakdown at 32 degrees centigrade. So it's quite a stringent standard to meet. And if that stability isn't met, we risk failure, so the infection doesn't get better. We perversely also risk, potentiating resistance because we're giving suboptimal dosing of the antibiotics. And in some circumstances, we're also infusing the breakdown products of the antibiotic, which might be harmful to the patient. So if we kind of just do a little bit of a stock take on, gram-negative infections in particular, gram-positives are actually quite a bit easier to treat on hospital in the home. And, what of those new and commonly used gram-negative antibiotics we can give, we don't have that many. Meropenem, which has been our kind of workhorse antibiotic for a lot of complicated infections, really isn't stable in this way. Some programs use it, but it's probably not optimal and not recommended. And of all of those kind of new agents I talked about, really, most of them are a big no, unfortunately. Cefidoracol, meropenem, vaborbactam, imipenem, relebactam, these are the kind of the three agents on that first list. None of them are suitable for, this intermediate space of hospital in the home care. That last agent, plasmamycin, that I talked about, is suitable, but has a lot of limitations about how you use it and what you can use it for. So, probably not the best agent to use. And in fact, if you look at our other kind of relatively recently, marketed agents for gram-negative infection, in fact, only one of them, ceftolozane tazobactam, which is used to treat resistant Pseudomonas infections, is suitable for hospital in the home use. So we really, whilst we do have a number of antibiotics coming to us to use for these, complicated infections, we're precluding, their use in this emerging hospital in the home space, which is challenging. So we've kind of hit this convergence of increased use of this space, but rising rates of resistance, really leading to a contracting scope of, the agents we can use in this space. So you can't really give a talk like this without proposing some solutions. And I guess, there's a couple of levels that we can really tackle this on, and some that I'm working on, and some that I hope all of you can work on as well. At a very basic level, I guess, in pharmacology and development of therapies, it would be great to, think about this use of the agents now that it's becoming more common when we're developing them, and thinking about the stability. You wouldn't invent, or you wouldn't develop an agent for HIV where it's all oral therapy, or TB where it's all oral therapy, and not be able to give it in the community. And, maybe we need to think about that with our antibiotics as well a bit more. One of the challenges for us is that, when these agents come to market, we actually often don't know if they're stable. And, for that one that is stable, ceftolozane tazobactam, it took us about 7 years after it came to market, but before we really had, definitive data showing its stability. So there's a huge gap from the time we get an agent to whether we work out if we can use it in this way. And finally, we probably have to accept a little bit, that some of these agents aren't stable in the traditional way we want to use them in this space, and we need to innovate around that, and we need to really translate around those difficulties we have with the agents. And that nanoparticle talk was really interesting because there is a lot of work on looking at ways to make these agents more stable by pairing them with some of these things. I'm involved in some work really looking at the way we give these antibiotics in the community and seeing if we can kind of support their stability by simple measures like cooling them down and keeping them cool as we give them in the community. And there's probably some clinical and human factors as well. And there are even programs as simple as trying to teach patients to give their own antibiotics at home. So even if the nurse can only come once a day, then the patient can give their antibiotics another time in the day. So that's really all I wanted to talk about. And I guess if I want to wrap it up, and say a few key messages from it, we do have a number of new antimicrobials coming to the market, which is good, not enough and not broad enough, but we have a few. For gram-negative infections, these really at the moment tend to be very much focused on the very pointy end, sepsis, sick critically ill patients in hospital, which doesn't really cover the full spectrum of AMR infections that we need to treat. The space I work in, hospital in the home or OPAT, really is a growing location for antimicrobial therapy and a need for, treatment of antimicrobial resistant infections. As I've said, a lot of the new agents aren't suitable for this. We need to focus on this in kind of the drug development space, and we need to kind of, innovate and translate to deliver our existing less stable therapies in this space. Thanks so much.
Has anyone got any questions for Ben? Anton.
Just interested in your thoughts on long-acting antibiotics and how you're seeing them fit in the system and whether you're using them and the potential benefits, pros and cons.
Yeah, so that's it. Oh, I think this one's working. Can you hear me?
I'll take that one back. You can take that one back, yep.
There's another one.
So I think, Anton, that's a really good comment. And, we have in some spaces antifungal therapy, gram-positive infection, seeing some incredible antibiotics that you can actually give, once a week. And, these are, something that we will really try and utilise in the OPAT space. But they really tend to be focused on very particular types of infections and won't meet our whole need. But they're probably a really good example of what we can do, to solve these problems.
Does this one work? Yes, it does. I guess the flip side to long-acting antibiotics is their very long half-life and their subtherapeutic tail, and the cautionary examples we've had from drugs like azithromycin and agents like nevirapine. So it's a really double-edged sword, isn't it? There's some enormous benefits, and then there's some terrible examples that sort of tell us that we might lose them fairly quickly unless we can buffer them in some way. I remember in the old days with nevirapine, we used to try and add combivir when they were stopping for like an imaginary 14 days, whatever it was, to try and sort of prevent that monotherapy. Any other questions? Yes, Trevor. Sorry, here's me holding on to the mic.
Katrina, could I just follow on from that point? One thing that was concerning my non-clinician brain on this was if we go for rock-solid carbapenems and other drugs that are long life, and then we let people go out in the community and wee and poo them all over the place, are we worried about the accumulation in the environment of these very stable drugs?
I think that's a huge issue that we don't really understand much about, we always think once the patient kind of, wheezes and poos out the antibiotic, it just disappears somewhere magical, but we know, that stays, in the environment. It ends up, in animal husbandry and things like that. And, that's a whole, space we need to think about in the development of new drugs. Yeah.
Yes. Sorry, Simon.
Thanks, Ben. I was just wondering, are there projects underway about formulation of these meropenem or other antibiotics specifically for use in these infusers? Because there's plenty of people around who might be able to do that.
Yeah, I think we'd always love to talk. There certainly are projects looking at, nanoparticle suspensions and things to keep them more stable. One of the challenges of that from a regulatory perspective is that you're really inventing a new drug, and it has to go through that whole new drug process, which is a little bit painful.
Yeah, I just wonder if you can stabilize it for the time that you need, and then deliver just the drug as the nanoparticles are great. Don't get me wrong, but as you say,
yeah, and look, and I guess we're working in that space as well, and we're looking, as I said, at really simple things like a lot of these drugs actually, if you keep them cool, are stable. So looking at ways to actually deliver them in the community. Cooled rather than at body temperature. But there's a lot of work in that space, and there's probably a lot of innovation outside of biomedicine that could be brought into it.
Cool.
Yeah, I would have thought keeping it cool is the least technically challenging aspect to the entire thing somehow. All right, so on to our next speaker, Sam. So many people here know Sam. He is a bit of a microbiota guru at the Hudson, but particularly focuses on the gut. He tells me he's not really doing much work on the lung, and we're trying to attract him into the vaginal microbiota space slowly, aren't we? So exciting. And so let's get this slide out of the way. And here we go, Sam. Over to you.
Thank you very much, and thank you for the invitation to speak today. So I'll start up by saying our knowledge of antimicrobial resistance is somewhat limited, but our knowledge of the microbiome is very detailed. So the perspective we come to this from is really understanding these bacterial communities that exist. Most of the bacteria we deal with aren't pathogens. You don't even care that you have them, but they do interact with the pathogens. And so that's sort of the perspective that I'm coming to this from. I should say I've got a few conflicts both with Biobank and Microbiotica, so don't believe anything I say. So I suppose this sort of gives the concept of where we're starting, right? So we know that there's a huge number of pathogens. We think of them from a human perspective because we're humans and we can care about things that are infecting us. We know that they spread antimicrobial resistance around through the environment, through agricultural settings, and we know that obviously everyone in this room knows that's important. From our perspective, we know humans carry a whole heap of commensal species that play a critical role in the health, but also interact with these pathogens. So the reason that I want to talk today, the things I'm going to focus on are really how are these bacteria interacting with the pathogens? How can we leverage that knowledge or the knowledge that we can gain from those interactions? And how can we come at this problem from a subtly different direction to perhaps the normal thought processes? So the first obvious point here is these commensals compete with a lot of the pathogens already. So they have mechanisms for killing them. And so the first question, obviously, that raises is what are these mechanisms? How can we understand them? And how can we leverage that knowledge to develop new therapeutic interventions? The second key element of this whole game is not only do they interact, they also share DNA, obviously. So they become a reservoir for antibiotic resistance. And they also are potentially a source of new antibiotic resistance that we're not even thinking about at the moment, right? At the moment, we're very reactionary in the way we're thinking about it. We see a pathogen, it has the resistance, that's when we care about it. Actually, there's thousands of species of bacteria that we're carrying already that do have resistance there and actually could transfer that to the pathogens. So fundamentally, the thing we're interested in is these interactions. How are they occurring? And obviously, how can we look at the movement of antimicrobial resistance from both commensals to pathogens and pathogens to commensals? And obviously, I'm biased by humans, but we should say every one of these environments has a microbiome as well. Same principles, same concepts, different species. And so it becomes very complex very quickly, but there's a huge opportunity here. Instead of reacting when the problem exists, we can start to understand the background, understand the biology, and understand how nature does it already and leverage that. Obviously, for the purpose of this talk and because of my bias, we're going to focus on the human microbiome and particularly the gut. And as you heard, my group really focuses on the gastrointestinal microbiome. Everyone, I'm sure, is aware there's a huge number of bacterial species that exist in the gut, right? So the density of those bacteria increases as you go through the gastrointestinal tract, but we're talking trillions of cells, millions of genes, depending on how you define species, hundreds or thousands of species. Each one of them has its own genetic composition, its own ability to interact with the pathogens, its own ability to share antimicrobial resistance with the pathogens. And so as I've kind of alluded to, the three key questions that I want to focus on today. The first is whether we can phenotypically and genomically characterize novel antimicrobial resistance within the microbiome. The second is, can we measure and quantify the transfer of antimicrobial resistance between pathogens and commensals? And the last is whether we can find novel antimicrobials within the microbiome. So the first question, as I said, can we understand these antimicrobials? So the first thing I should say, and this is work from a few years ago, so I won't dwell on it, but a key element of this is we can grow these bacteria. So I know a lot of people say still that the gastrointestinal microbiota is unculturable. You can grow it. as soon as you can grow it, you can phenotype it. You're not completely dependent on genomic prediction. You can also go and test and see whether it's actually functional or it's actually able to perform as you're expecting from the genomics. We now, the other key point, we now have large collections of these species. So it's not just a genomic sequence that we're interrogating, it's an isolate we have in the freezer. We can go and phenotypically characterize what's in there. So that gives you a context for what we're focusing on here. We can grow these bacteria, we can genome sequence pure isolates, we can understand what's in there. And then when we start looking at these isolates, we can start to say, well, what's in there? And the first thing we see is pretty much every species phenotypically is resistant. So it's not a characteristic of pathogens. It's something that's there for all of these species. Depending, obviously, it's not universal. Depending on the profiles, depending on the species, rather, the profiles are different. But fundamentally, there's resistance already there. And so the key problem we have, and I realize it's supposed to be scientific solutions here, but this is scientific problems. The key problem we have is we can't predict this antimicrobial resistance from the genomes. So we know it's there. We know phenotypically it's happening. We know in some cases it can be transferred, but we can't predict it. So there's a huge gap in our knowledge here that needs to be filled. And obviously, the second half of this question, which I'll get onto, is how much of this can then move into pathogens where we actually care about it. So that just gives you a starting point in that space, but it shows you phenotypically we can find these interesting things within the commensals. The second question then becomes, well, can we measure and predict the transfer? So, okay, the commensals are resistant. Who cares? That might actually be beneficial, right? You're giving people antibiotics, they're resistant, it doesn't affect the commensals. But if they can be then transferred to the pathogens, that becomes a problem for us. And so this is some work from quite a few years ago now, but it is historically where we started in this space. So that's the way I'm going to tell this story. So C. diff and the microbiome, this is work when I was at the Sanger, and we had a large collection of C. diff isolates, and we just asked the question, well, how frequently do we see resistance from C. diff shared with genetically with the commensal species? And you can see here, every one of the around the outside is a phylogenetic tree, and every one of those lines is a point where we had high genetic similarity between an 078 strain of C. diff, so not one of the more pathogenic strains, but a 078 strain of C. diff and commensal species. And obviously, you don't need to look at that in a lot of detail to see there's a lot of shared genome content. And obviously, we can then build elements around this, and I'm not going to go into the detail. If you care about the detail, obviously, you can look in the paper. But the key point that this allowed us to do is we could start to see elements that were shared between C. diff and the microbiome. And the nice thing about this, obviously, is because we've got the species, we can then go and look at them, and we can characterize the resistance specifically. In some cases, though, genetic modification of these species is challenging. We can obviously do traditional microbiology to validate the phenotypes. And of course, once you've seen that once, you go on a broader level. So we then took over 1,000 commensal genomes, a large collection of pathogen genomes, and you can see the species in that first figure there that were actually represented, and said, can we do the same thing? So we're in this phylogenetic tree where you're seeing those red dots are the pathogens, and where you're seeing the lines, that's the species that we're seeing shared antibiotic resistant gene plus a surrounding region, so suggesting transfer. Again, you can see a large number of lines, but actually the thing we were most interested in, and the thing I should have drawn your attention to already, the majority of the commensals aren't actually phylogenetically that close to the pathogen. So the other way of thinking that, the pathogens are rare. So as a result, the elements that matter the most in this context are the ones that can go a long distance. So we can actually ask that question quite easily, not with this visualization, obviously, but we can start to say, well, what elements are we seeing that are actually able to move large distances? And unsurprisingly, the elements that move a long way, much rarer, but they're there as these broad range elements. So these are elements moving between bacterial phyla. So what we're able to do is we're able to say there's a huge number of elements. Some of them matter more, and that's these ones. And then we can go, I won't go into this figure in detail, but you can start to see how different pathogens carry some of these elements that we're identifying, and then which commensal species they're sharing them with, and obviously other characteristics of these elements. So we can then start to really understand the nature of these elements, obviously, because we can genome sequence them, etc. And more importantly, we can actually go back and experimentally validate. So we can take these isolates because we've got them in the lab, and we can put them together, and we can, for example, here, we're seeing species transferring antibiotic resistance to Fecalis just by a co-culture experiment. So previously unknown mobile elements, but by discovering them through these genomic approaches, we can go and identify them, and then we can go back and experimentally validate. And obviously, this could be happening in someone's gut. It could be happening in the environment as well with other species. But it gives us a way of an unbiased approach to finding these relationships. And so, obviously, from a mobile element perspective, that's the second part of the puzzle. We can say in the sample we have these species that are carrying the antibiotic resistance. We have an element there that's not just moving to its neighbor, but is moving it to some pathogen in a different phyla. They're the elements that gives us an ability to start to prioritize the importance of an antibiotic resistance gene in the context of a mobile element, in the context of a species, a commensal species. And that, I would argue, is a huge opportunity to start screening patients and screening people to understand where these events might happen before the resistance is in a pathogen and you're treating a patient. And just to point it out, so I do some vaginal microbiome, I will point out, at least on the informatics perspective. We can start to look for some of these common elements in various environmental contexts. And obviously, as you can see here, the these key elements that we know are broad range, unsurprisingly, they're broad range, they get into lots of environments as well. So it's stating the obvious, but it's a way of us being able to find them and prioritize them. And then the last question I realized we're on a bit of a tight schedule, but I wanted to focus on was, can we find antimicrobials within the microbiome? So it's one thing to say, well, there's antibiotic resistance. Well, of course there's antibiotic resistance. There's antimicrobials being produced there as well. If any species is going to exist in this environment, it needs to be able to protect itself from the other commensals that are producing antimicrobials. And obviously, from our perspective, pathogens are the same as commensals. They also live in these environments. They also interact in these environments. So the question becomes, well, can we find novel antimicrobials within these species? And one could argue that actually this is one of the most sensible locations to look for antimicrobials, because a lot of the pathogens also exist in the gastrointestinal tract. So it gives us this opportunity to say, well, and again, based on the ability to grow these species, if we take these commensals and look at their ability to interact with various common pathogens, what do we see? And the answer, as you might expect, is there's a huge amount of resistance, a huge amount of inhibition that is able to occur in the presence of these commensals. And that gives us an opportunity to start to understand these resistances, as these antimicrobial production, and the resistances within in pathogens. So what you're seeing here is just a few examples just to highlight a couple of key elements. Obviously, if we take commensal-like things and put them through these assays, number one, we see easy or relatively easy to predict antimicrobial resistance genes. But when we take a lot of the commensals, we've got no idea what these antimicrobials are. We can, and I'm not going to go into the detail today, but we can start to obviously understand the type of molecules they are and these types of things, but we can't genomically predict them. So we can't take the genomes, all the genomes we have from the commensal microbes or any of the other microbes for that matter, and know which ones to work on. But with these high throughput screens, we can start to identify candidate species that are really worth focusing on. And really worth understanding. And that then obviously takes us to the next question, which is, do we even need to make them into antimicrobial molecules or antibiotics? Do we actually just try to modify the environment? I think historically as humans, we're pretty good at eradicating species, and that usually is dependent on destroying environments. So maybe we take the same approach here. We can start to modify the environment where these pathogens exist, and make it less likely for them to exist for long periods of time, and therefore less likely to be able to take up antimicrobial resistance, but also less able to exist in general, so less chance of exposure. I'm not going to go into the detail of live biotherapeutics today. I'm not going to go into the detail of fecal transplant, but fundamentally, there's lots of different ways we can seek to modify these communities. And the question is, can we do it in a targeted way such that we reduce, for particular patients and particular people, the risks of the infections happening in the first place, rather than just responding and reacting once they occur. So I think I've said this, but as I said, we really focus on the microbiome, the gut microbiome. There's a lot of evidence now of this as a source of antimicrobial resistance, but we're actually starting to get to a point where we can understand both the genomic composition, but also the phenotypic capabilities of these species, and we can start to understand how to target particular isolates or species or strains within these communities where they're likely to have a more significant effect than others. As you probably gathered, the informatics here is still lacking. We know a lot more phenotypically than we do computationally, though I think sometimes we fall into the trap of thinking we can predict everything from a genome. The reality, and as a bioinformatician originally, I have to say I wish it was that way, but it definitely isn't. But as we start to build these databases, we can start to reanalyze existing data sets using this new knowledge to uncover new relationships. So thank you everyone for listening, and obviously there are a lot of people involved who I'd like to thank also. So thank you very much. We can share one.
Thanks very much. That was a fantastic talk. I really enjoyed this. So I have a question. Obviously we have just woken up as humans to this struggle which has gone on for millions of years, right? What do you think about resurrecting previous, candidates that have been, basically overcome by bacteria and other microbes? Is that a worthwhile area to search for as well?
I wish I knew the answer. I think at this stage we're still at the point of discovery. So there's so much out there that because we focus on the patient as we should, that has the infection at a particular time. The whole history of why was that pathogen, how many people have thought when they're treating a patient, where did this pathogen get this resistance from, 3, 4, 5 million years ago? No, unlikely. How many thought about it the other day when that person was in the environment, sharing? We've got to that level of thinking. So I think we're moving that way to really understanding the relationships. But the challenges of how do you prioritize, that's a question for someone who understands these things much better than I do.
Great talk, Sam. I must have missed this. So, what's the criteria for you to determine that the commensal had antimicrobial properties?
Yeah, sorry, I jumped over that a lot. It is actually on bioarchives now, and I should have put the reference up. But basically, there was a whole series of initial screens, which were just overlay assays to wipe out, because we've got 40,000 isolates initially, and that was to prioritize. Then we went through a series of tests with different treatments of both supernatants and the isolates, whether heat killed would kill, all of these things. So there was a series of steps going through to rule out the obvious, pH changes, these types of things that you'd be worried about.
Great talk, thanks. I was fascinated by your last slide, which I think was bioengineering a microbiome. How would you choose a candidate?
Yeah, I mean, there's I suppose a lot of companies at the moment playing that game. I think there's two elements to it. One thing I'd say is it's not going to be a single candidate in most cases. You're bioengineering, you're changing the whole community structure there, right? And so the stages you're going to have to go through. The first is disrupt the current state, because obviously even in a disease situation, you have a stable microbiome state most of the time. It's difficult to move away from that. So you'll have part of the mixture you'll disrupt that community. The second element, you need to create a state where your actual therapeutic candidate is able to get in there and exist. So the equivalent, if you were imagining in a macroscopic world, you'd be planting the right trees to allow your species of interest. And so it really is, it's that level of community understanding and interactions within a community to say, this is a state that excludes certain species and promotes the existence of others. So, and some of those will happen naturally, right? If a species is already producing an antimicrobial to kill a pathogen you don't want, the likelihood is that there's a reason that species has evolved to produce that antimicrobial to kill the pathogen, so they probably don't get on. So then it's likely there's a community state where you can just encourage that behavior effectively.
And how would you choose the person?
So computationally, you can do that relatively easily. You start with. I'll keep talking because I can. Basically, you start with the tens of thousands of isolates you could have. Sorry, do you want to?
I was going to ask you, how do you choose the patient before they become a patient of AMR?
Sorry, I'm going the other way. Yeah, so I think, I don't care about patients, I would say. But
exactly. So how would you choose a community person for this?
So basically, you're dealing with high risk individuals. So anyone, the argument could be that everyone.
Yeah, exactly.
Why not? As in most people, if we go back, I'm not sure if I can go back or it'll stop me. These are the ways you change the microbiome. Most people, I don't think I'd ask anyone to change their diet, but they could at least take a probiotic with their food. So you can actually already modify the environment relatively easily. The question is how many people would want to do that? Well, if you were saying it reduces your chance of infections, if you felt you were susceptible to that infection, I think a lot of people would. You look at the take up of probiotics, which have questionable efficacy, and every second person seems to take them. So I think there's opportunities there. Just you don't actually need to treat the patient when they're a patient. You can be preemptive, and effectively you could create an environment where the pathogens can't exist because there aren't enough hosts for them when they're at the commensal stage rather than when they're infecting.
Do we have Philip here? Oh, excellent. Good. That's great. Okay, so we've just got time for one more question and then we can go on.
Sam, really interested, in this the concept of preventing colonization or carriage of the pathogen, and how the gut microbiome can prevent that. But also interested to think not only the commensal and its effect on the pathogen, but also the commensal in terms of the health of the gut, the mucosal aspects, mucosal epithelial aspects, the immune response. And I'm sure the commensals are really important in gut mucosal immunity, anti-inflammatory properties, and just health on the host mucosal side that could prevent and support resistance against resistant AMR.
Yeah, I think there's one comment I would make at the start is the distinction between pathogen and commensal is a difficult one before we start, right? And so I think we should start from that point. But absolutely, we let the commensals. I mean, let is probably the wrong way of describing it. But we have immune systems. We could get rid of the commensals if they weren't beneficial. And so we've played this game effectively evolutionarily, where our immune system is willing to tolerate some bacteria, even at the risk of some of them becoming pathogenic. So the reverse of that argument is obviously that there must be a benefit that's stronger than the risk of having the pathogens to having these bacteria around. And as you say, all of these host modulation type elements come into that. And
would that be important when selecting a live biotherapeutic product, for example, that you'd want to not only just look at that, but look at effects?
Yeah, that comes back to the point I was making was around having the community state right. Because, I mean, the other thing we have to remember is just because it does it in one scenario, it doesn't mean we're going to be able to induce that state. So you actually need the community state of the bacteria to be right for the effect to happen. Thank you.
Great talk, Sam. I will hold over some questions to the panel because I think we all have lots of questions. Philip, sorry, we've been running over time. Let me just... We have to actually locate your talk. It is... Let's just find it in this folder, I think. Now, where are you? This one? Yep. Excellent. Yeah, looking good. All right, so we'll do a very brief introduction. Philip is a chemist, and we're going to be learning about metal-based antimicrobials, which is a nice change from drugs. So I'll hand over to you straight away.
Thank you very much, Katrina. So I guess this is the point where you either get inspired or you have nightmarish flashbacks to having studied undergraduate chemistry. Because I'm going to talk a little bit about chemistry and I'll try and make it as clear and as understandable as possible. What I'm going to do is probably let you know a little bit about why we study metals in terms of their antimicrobial properties, and then talk to you a little bit about the difficulties and the challenges of dealing with metals, because it's clear to anybody that while there's great potential and metals have been used for thousands of years as antimicrobial disinfectant agents, that these days on the market, the pharma industry is still not particularly interested in pursuing strategies around either medicinal metals or metals in disease or even in antimicrobials. So I guess what I'm trying to do is be a little bit of a sort of evangelist in terms of trying to convince you that there's a place for metals again and trying to solve some of the problems we have around AMR. And hopefully, by the end of it, and I'll give you 4 examples of our own work where we think we've made some significant progress. It's, of course, still at a very discovery stage, that there's potential in taking this forward. So you've just read the screen where I'm thanking some of my high-level collaborators that we couldn't have done the work without. You see from the collaborators at the bottom, while we're chemists, we collaborate with engineers and we collaborate with microbiologists as well. So in the last few years, and you can simply pull this back to the fact, if you look at the antibiotic pipeline and the number of new antibiotics that are coming through, everybody knows the data. Normally I would show it. It's drying up, it's becoming difficult, and we know that it's very difficult to beat evolution. Doesn't matter how hard and how fast we run, we always seem to be now behind the 8 ball after 80 years of sort of antibiotic panacea. So people are starting to look back
at what has previously happened. These are papers that have came out in the last 2 or 3 years. In Australia, driven by the group up at the University of Queensland, who were running the COAD group, and if you knew anything about them, they would simply take compounds from anybody, anywhere in the world, and they would assay them against some of the escape pathogens, excuse me. So what they did was they collected a whole load of data from metal-based compounds. They've published it, you can read it. And what they've shown is that from this diverse group of complexes, excuse me, you can see that there are metals across the periodic table which will act as antimicrobials. I guess the biggest criticism you could throw at them was it wasn't a very systematic study. It simply takes all the compounds that were submitted to them, and they've gone, manganese can act as an antimicrobial. Ruthenium can act as an antimicrobial because here are compounds that actually do it. And the way that metal compounds work, that approach, the suck it and see approach, doesn't really work very well. And it's part of the reason why the pharma industry is generally quite cold on the development of metal-based therapeutics. And I'll try and explain that as well as I go through some of these slides. So here are the metals that have been discovered to have antibiotic or antimicrobial activities. You'll see the top row, the transition elements, of course, are the essential metals. So they're largely endogenous metals. They've evolved roles within our bodies primarily through enzymatic functions, and therefore some of the targets of metals are clearly going to be enzyme disruptor. But you also get other aspects that metals play within the body, and they're multi-targeting and they're multifunctional. So when you think of small molecule antimicrobial development, by and large, they're single molecule, single function, single target. And what you do is you take a medicinal chemistry approach and you change a methyl group for a trifluoroethyl group, or you change a chloride for an iodide, and you look at the change in function. Now, when you have one target, evolution runs past you because you develop genes that can get past that. When you have a multi-targeting approach, so whether you're disrupting the cytoplasm or the membrane or enzymatic function or oxidative stress within the cells themselves, then that's great because you're not limited to one target. But when you try and design a metal-based therapeutic or antimicrobial, the fact that it's multi-targeting introduces a whole load of problems because it's much harder to pin down where the actual function lies. The benefit is, of course, if one of those targets is bypassed evolutionary, there's probably 3 or 4 other ones that are going to be residual within it. Now, our interest lies over here in what are called the main group elements. We look at some transition elements, but not a lot, and I'll tell you why that's the case. Now, there are metal-based drugs, of course. The top one, cisplatin, is the most common anti-cancer drug that's used. It's incredibly toxic. DNA intercalator works very well, but as everybody knows, it makes people also very sick as they undergo the chemotherapy. There are other ones, sodium stibogluconate in the middle is an antimony-based compound that's used to treat leishmaniasis. It has been for the last 70 years, and it's still used as a frontline drug. Other metal-based compounds, particularly ruthenium anti-cancer drugs, some gallium anti-cancer drugs have made their way to phase 2, phase 3 trials. Not many make it through into the clinic, unless you're looking at imaging agents where you've got very stable gadolinium or other rare earth or radionuclide centers. And there's reasons why those are very, very stable. And there's mercury, of course. Everybody used to love a bit of mercury on their wounds and in their gums when they were having a bit of a problem. So our main metal antimicrobial, you'll know at the moment, is silver. It's everywhere. So you go into the local store and you can buy Band-Aids impregnated with silver. There is something about silver which is really interesting. Of course, it's been known for thousands of years. Used to be added to water in ancient times as a disinfectant, as a purifier. And even recently it was added to wine to help take out sulfides and also keep levels down without any preservatives. So silver is well known. It works very well, I think, outside the body. I don't think there's much evidence that in vivo it's actually very effective. And that's primarily because it floats about as a silver ion and it migrates very easily. It looks a bit like sodium, potassium, and therefore efflux pumps have developed that will simply pump silver away from cells. So it's very good. There are, of course, problems with these. and is that now the widespread use of silver into the environment is starting to generate some resistance. It's been recognised, and it's probably not the best material that can be used, but it is effective if it's it's going to be used on your skin, or even within plastics. It's not particularly effective. So we need alternatives to that. Now, this is some of the work that we do. What we look at metals which mimic iron within the body, particularly iron 3. And so we're interested in metals which are very closely chemically related with iron. Why is that important? So if you think about a bacterial cell that decides it's not going to let a metal in, an exogenous metal, which is carried, looks like iron, is carried by the same proteins, uses the same transport mechanisms, is taken into the cell by the same mechanisms. If it suddenly decides, whoa, that's not iron, and it starts to close down those mechanisms, well, it's sort of digging its own grave anyway. And so it's very hard for microbes to develop processes that block out the use of metals inside their cells. They're so critical to the function of them that if they start to close down those pathways, then they're going to bring about their own end anyway. So evolutionary wise, it doesn't make sense. So metals that mimic iron are very important. We look at gallium, and if you're interested in the chemistry, gallium is very similar to iron. I can't go into it in great detail, but if you look at the biochemistry of gallium, it essentially follows iron around the body. The body finds it very hard to distinguish between the two metals. We also look at bismuth, which has got different chemical properties. Let me say, in chemistry terms, iron and gallium like to bind to ligands, and these can be proteins, which have got oxygen and nitrogen binding sites. Sulfur is more relatable for bismuth because it's a heavy metal. Heavy metals tend to bind to sulfur, which will be in cysteine or in methionine residues. And so they get shuffled around, very much like mercury or lead or any other heavy metals that are there. So the chemistry of iron and gallium are very similar. So we need to investigate whether the one property that distinguishes them in the body, and that is that iron has a redox role. It flips between an oxidation state of 2 and an oxidation state of 3. 2 generally transports iron, 3 generally stores it. But it's also critical for a lot of enzymatic processes. Gallium has no redox chemistry, and therefore, while it might look like iron, it doesn't behave like iron in the body. And so cells have got a real problem with dealing with it. And a few gallium compounds have made it their way through to phase 3 trialing as anti-cancer compounds. So why bismuth? Bismuth is known as a non-toxic heavy metal, and these days I put non-toxic in those air things because we've done a lot of studies to show actually it can be quite toxic to cells. But generally, by and large, as this paper in Nature says, many of the bismuth compounds that have been trialled within medicine and used have got lower toxicity than sodium chloride. And so as a heavy metal, you wouldn't do that with mercury, you wouldn't do it with lead, you wouldn't do it with thallium. Funny enough, bismuth has this really unusual property, is that when it's taken up, it's got very low systemic toxicity, and when you stop taking it, the body gets rid of it. And so you actually regain the function. You don't do that with mercury. If mercury gets into your brain, you've got that problem forever, and you might die with it. And so it behaves completely differently. This is a recent paper that looked at bismuth antimicrobial drugs. So this is really interesting for us. Hongzhe Sung co-administered bismuth citrate, and it knocks out the metallo-beta-lactamases. And so what it allowed was those beta-lactam drugs continued to work when they were co-administered with bismuth citrate. That's because bismuth itself will exchange with certain metals within enzymes and deactivate them. And we know that's the case. So it's an interesting metal for us to look at. Of course, because of Warren and Marshall, everybody knows about bismuth in the treatment of Helicobacter pylori. One of our collaborators, Richard Ferrero at the Hudson, of course, looks at Helicobacter. So we started looking at bismuth compounds and the effect they had on Helicobacter. Hongzhe's son, who is in the University of Hong Kong, has looked at the role that bismuth tends to play in targeting Helicobacter. And when I talked about multi-targeting, this is multi-targeting, right? So it inhibits the ureas. It disrupts alcohol dehydrogenase, it's cytoplasmic problems, it's membrane disruption, ATP. And so you can see that when the metal goes in there, it's just going bang, 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 bang. It's like a cluster bomb. And so that's very difficult to control, but it's incredibly effective. And as we know that people have been taking bismuth-based drugs to treat Helicobacter, they never knew it until Warren and Marshall showed them, but for over 120 years. Because Pepto-Bismol, which you can buy in the States and in Europe, you just drink it, and you drink as much as you like. The worst thing is your tongue goes black and your stools go black, but it was for the treatment of gastrointestinal ailments. And it's one of the main treatments now in quadruple therapy, where other drugs like clarithromycin, metronidazole have now got high levels of resistance. It's now used to knock out Helicobacter, and it does it in a week. In 120 years, no resistance has developed to bismuth in the treatment of Helicobacter. Oh, sorry, I jumped too far. So this is some bismuth compounds. I just want to show you how effective they are. It's just an illustrated example. The medicinal bismuths tend to be derived from carboxylic acids. We've looked at a whole range of different functional groups. These are sulfonates. We looked at sulfonates because they've been studied at a high level by the Europeans, so regulatory processes. Because they're used in everything as surfactants, sulfonic acids are generally treated as non-toxic entities. And so we're interested in non-toxic entities to put our metal on because it knocks out the effect of the ligand by and large. And bismuth will take it down to nanomolar activity against Helicobacter. To put it into context, I don't know if it's there, it's not. Bismuth subsalicylate works at about 12 micrograms per mil. So you're orders and orders of magnitude more effective by changing the compound you're on. If we go away from Helicobacter, we can look at other bacteria. The gram-negative at the end, you can see E. coli. These compounds, which were membrane disruptors, we link to bismuth and we get fantastic activities, low micromolar activities across the spread of bacteria, except the gram-negatives. The gram-negatives are really often resistant to metal-based compounds, and I think it's the double membrane that causes a bit of trouble with that. But we can get through them as well. This is a bismuth phosphonate. This compound is derived from a phosphinic acid, and they're incredibly insoluble. So 1 gram of this compound takes 16 liters of water to dissolve it. So it's fantastic for putting into materials, composites, polymers, which are another area of the work that we try to do. So I want to point out an important thing here and see if the chemistry goes this far. This is what we call a heteroleptic compound. It's got 2 types of ligands. It's got an aryl group, which has got a carbon-bismuth bond, and it's got a phosphonate ligand attached to it. And so there's 2 types of ligands. And these metal-carbon bonds and the heavy metals tend to be what we call non-labile. They don't exchange very easily, so they're stable. So when we acid this compound, we stab it because we can't dissolve it. It must have some solubility in the agar because it gives you diffusion zone. What if we go to what we call homoleptic compounds, and you can see now it just has 3 phosphonate groups around it, is that there is no activity against any of the bacteria whatsoever. So simply swapping 1 ligand out for a phenyl group, which has got a metal carbon bond rather than a metal oxygen bond, increases the antibacterial activity phenomenally high. So tris compounds don't work particularly well. We put that into, with our colleagues in Bioprea, we put them into paper. We've looked at them as filter papers that filter out microbes and kill bacteria, and they work effectively well. This is our comparison with silver sulfadiazine. This is the important levels. This is 0.5 weight% of bismuth, which would be below a regulatory threshold, and we outperform silver very easily in these things. So this is why it's a very effective material. So against everyone except E. coli, it's always a problem. We do that. This is when we put them into hydrogels. So this is the toxicity of our bismuth phosphonate loaded into a cellulose hydrogel. And you can see at this point here, which is a sweet spot of about 9 micrograms per gram, we have full mammalian cell viability. If we compare it with silver, silver is much more toxic, so our compounds are much less toxic towards the mammalian cells than the silver equivalent is. So if I jump from there to a live dead cell assay, this 9 micrograms per gram, which is over here, is the sweet spot for where we get a high selectivity between the mammalian cell and the bacterial cell activity. So we took that and we looked at the effect of these gels, and you can see that against A. baumannii in 4 hours we can knock it out completely with the gel. We can do the same with Pseudomonas. It takes a bit longer, and the other ones we get a bacteriostatic response. And so it stops the growth essentially of the bacteria. And at the moment we're looking at those gels as wound coatings. Last 2 examples. Flavonoids, which, as you know, if you have a good diet and a good microbiome in your stomach, come from vegetables. And so they have some antimicrobial activity, they have some anti-tumor activities. We derivatized them with bismuth as a non-toxic, safe, acceptable ligand. We want to know what the bismuth does. So here we go against a range of bacteria, Staph aureus, MRSA, we've got the gram negatives. And if you compare just the flavonoid itself, does nothing. If you have the homoleptic, so it's got 3 flavonoid ligands around the bismuth, you get some antimicrobial activity. If you have a heteroleptic compound, then you've got better activity. And this time we're seeing activity against E. coli, the gram negatives, which is really good. This is a point which I'm going to have to make, is that after our discussions with Claudia, who's here when we were doing some antifungals, she looks at iron homeostasis and the effect of iron. It suddenly became clear to us that because gallium is like iron, they exchange. So if I put a gallium compound into a solution with an iron compound, the iron 3 will exchange with the gallium 3 to a certain extent till it equilibrates. So if you try to put gallium in an iron-rich environment, the chances are you're kicking the gallium out and you're being left with an iron compound which might not have any activity. So if we now do these in RPMI with some bovine serum just to dope it with some iron, we get even better activities. And so this iron poor environment, which is much more physiologically relevant than using LB broth, becomes really important in the effect that they have. So here's now our gallium. So gallium compounds, here's our homoleptics, here's our organometallic gallium compounds that can't behave the same as gallium does. So on transferrin will take up gallium very easily. And it does it because it's a gallium 3 ion. It looks like iron. If you have a dimethyl gallium entity, the gallium carbon bonds don't exchange, they don't break. So transferrin cannot take up that entity the same way it takes up gallium, because it doesn't look like iron anymore. So it has to have a different mechanism, a different mode of action. So what's the activity of these? So we've got our organometallics against mammalian cells. So these are mouse fibroblasts, non-toxic, which is good for the tris ones, the homoleptics. The organometallic ones, a little bit more cytotoxicity, but not much, all within a reasonable state. And then we did the assays on the gallium compounds, and students came back and they went, they don't work, they're rubbish. And there goes your ARC grant. So we thought we have to reinvestigate this. So the whole thing was what Claudia told us, this iron poor environment is absolutely critical for understanding what the gallium does. So here we do it in RPMI, we do it with some FBS, and we've done it with human serum as well, just to dope it. And you go from being relatively inactive to being incredibly active. So in a more physiologically relevant environment, the gallium becomes much, much, much more active. And I think that's a fantastic result. And you can see that the compounds themselves are essentially non-toxic to the mammalian cells. So the organometallic compounds against Klebsiella are now in nanomolar activity. simply because you've changed the culture medium you do the assay in. How many results are in the literature where they don't think about the culture medium in which they do their assays? And we know that culture media is full of things like phosphates and amino acids and sugars, everything that will exchange with ligands on metal complexes. So, big breakthrough. We know using RPMI is the way to go. We did some ICP measurements. There's about 2.5 micromolar iron in RPMI. We add human serum, it goes up to 6. In LB broth, it's almost double the amount of iron. So the iron concentration within the broth has a big effect. And so we need to consider that when we do our assays. This is the last thing I want to tell you. It's our most recent breakthrough. So we've taken fluoroquinolone, the resistant strain, which is really a reference resistant strain, KP1074. We've made dimethyl gallium derivatives of the fluoroquinolones. The fluoroquinolones, of course, are not active at all because the KP is resistant. What happens when you add the gallium? So I won't hold you in suspense, is that here you have the gallium compounds, here you have the fluoroquinolone, doesn't do anything. The gallium ones are now nanomolar activity. We have reactivated the fluoroquinolone to actually knock out the Klebsiella resistant strain. So thanks to Anton and others, we're now looking at a whole suite of other drug resistant strains, not only of Klebsiella, but of AB and Pseudomonas. And hopefully we're going to see the metal activation is going to bring some of these drugs that have been long forgotten in use back into the game. How it happens, not sure. Anyway, that's me finished. I'm sorry if I spoke for a little bit longer. And I'll leave you with this, which is our spread that we got in Chemistry World when we described some of our chemistry with them a long time ago. Thank you, Katrina.
Thanks, Philip, for a really fascinating talk. So I'll just do a little bit of housekeeping. I can see Nanad looking anxiously at me going, what are you doing? So we're going to reconvene at 1:30, and I think we want to get people out at 4:30 to avoid the crush in Parliament Station. So what we might do is we'll take questions till 10 to 1 and have should we do that? A 40-minute lunch? So rather than a formal panel, we'll just have time to ask everyone questions. So if you want to all come up, I'll sit out of the way on the end here. First of all, any questions for Philip? I'm sure Anton's busting with questions. I have one question. How are you going to give gallium to patients with a systemic infection like Klebsiella pneumoniae? What's the proposed delivery? I mean, we all came from the sort of bismuth, swallowing the bismuth. Obviously, that's not going to be effective.
So the vehicle, the problem at the moment is that when you've got things which are largely insoluble, you can formulate them as solids. That then we do a simple test where we look at the compound stability against 0.1 molar hydrochloric acid. So that essentially, what happens if you put it in the gut? Does it just fall apart? Bismuth compounds tend to just fall apart, which is why they're used in Pepto-Bismol. Yeah. Gallium compounds don't. The organometallic compounds don't. So if you can engineer them to be largely insoluble, then you could theoretically take them in if they were lipophilic enough to cross the epithelium and into the bloodstream. If you've got something which is water non-soluble, then you're going to have to look at a vehicle that would be IV or intramuscular. If it was straight into the bloodstream, then it would have to be IV. But I'd have to say we simply look at the stability of these things. We would need to really be discussing with formulation chemists about how this goes.
That big next step. Any questions? Yes.
Thank you. Yeah, it's a question, Philip. Just being the devil's advocate, there's a lot of literature showing that metals have antimicrobial effects in vitro, but not a lot of them translate into clinical studies, and there's limited clinical studies. So how is this, your gallium's different to the others?
Yeah, it's a good question, and it goes to the fact why I think that the pharma companies don't particularly like it. So if I take you back to, say, organic derived synthetic organomolecules, which are antibiotics, or naturally extracted antibiotics, or peptides that are developed specifically for the purpose, they tend to be very stable entities. The way you develop a metal complex, as you've seen from this, is you tend to have a metal center, and then you have to stick things on it. The great thing about it is that you can stick things around it, and a bit, like a plug and play, you can take things off, put another one on, the problem you have a problem of thermodynamic stability is how stable is the complex you make. It can be stable as old boots, but as soon as you put it into a biological environment, those ligands start to exchange. And so the benefit of being able to design infinitely the structural chemistry around a metal complex is really intriguing. So it allows you to do the medicinal chemistry. At the same time, how do you control the exchange or the lability of the ligands? So if you take the gallium maltolate, which was in phase 2 trialing as an anti-cancer compound, Bernard Kepler's group, it got through to phase 2, and believe it or not, it was too stable. And so it wasn't actually shifting out its ligands to be able to get taken up into the cell effectively enough. And so it really comes down to the design feature of the complex you want. And I think for decades of medicinal chemists trained in organic chemistry, dealing with metals is a difficult thing, right? And that's why we need more inorganic chemists.
Anton, I'll give you this. The lesson from today is drug discovery is hard, hey?
Yeah. So, Phil, I was interested, so gallium nitrate, that's was used and I think approved for hypercalcemia in cancer treatments and things. It looked like it had great effects against, Pseudomonas and biofilm penetration. Now, I thought there was even a trial that's been done in CF with gallium. Is that gallium nitrate, and how's that different to the gallium molecules you're talking about?
Gallium nitrate is highly water soluble, which is good. And it was mainly used for bone imaging, right? For bone tumor imaging as well. You load up the body with a high amount of it. You can get up to about 1 micromolar circulating in the bloodstream without any toxic effects. That's a good thing. The problem is it clears very quickly as well. So it goes to what's called a gallate, which is even more water soluble, and it's simply excreted very quickly. So you need to continually load up the body with gallium nitrate simply because of the excretion rate. The way you slow it down is by changing the ligands and not just having the nitrate. So I think it's very effective. You need a high load in the gallium, but because it's short residence time in the body, it's very difficult to get where you want it to go, and it's hard to keep it in play within the body. If you do it in a in vitro study, it works very well, actually. And we've shown there's even gallium oxide, gallium hydroxides that work very well in vitro. And so gallium nitrate has been studied extensively, but it hasn't made the breakthroughs that people wanted. And I think it's that clearance rate that's the problem.
And it's interesting, Ben brought up cafetericol, which is a new cephalosporin that looks like a siderophore. So there's active uptake system, you get high concentrations in that periplasmic space. And what's interesting, your comment about development of resistance mechanisms, we're starting to see cafetericol resistance. And it's not just attacking the beta-lactam, so i.e. upregulation of beta-lactamases, they're starting to see the bacteria developing ion uptake mutations, which is really fascinating. So it's a combination of ion uptake mutations and upregulation of beta-lactamase. It just shows how smart these bacteria are that we're up against. Yeah.
You can keep the poll.
And given the huge efficacy you see going from LB to RPMI, I was actually wondering if you had thought about some adjunct therapy with something like deferoxamine, for instance, or if you can push the MICs to even lower levels and also reducing both of the drugs, the chelator and your gallium compound.
It's a good point, and we haven't really thought about it, and we haven't done it But given that you helped us with the RPMI story in the first place, then I'm really interested to look at how we manage the gallium ion concentrations, especially if you're going to move from being able to, I guess, synthetically control those concentrations to what you would be like in an in vivo situation. How we manage the ratio. The good thing is that, free iron in the body is not common, it's toxic. And so we would think that by and large it might not be an issue, but if siderophores, I think, are going to take gallium up quite easily as well, and we would have to look at the exchange rates and the stabilities, I think, but maybe we should talk what we do, Claudia. Yeah.
That sounds ominous, doesn't it? I think we've got Ned has a question.
Thanks, Katrina. Yeah, this is a question for Sam. So, great presentation. It was interesting to see you make that transition from microbiome to resistome, and it's something that we've been thinking about, especially with new sequencing approaches, that we can actually track the mobile genetic elements better. And we found recently that a lot of the kind of multi-drug resistant problems that we've run into have been due to plasmid transfer, in particular, across genus between different strains. Are you aware of any efforts at preventing that kind of mobile genetic element transfer, whether at the plasmid level or at the integron level?
Yeah, I mean, it's a good point. I think it's very early days for those types of interventions, but you can imagine mechanisms to actually stop plasmid transfer itself. The challenge there, of course, is there's lots of different mechanisms by which, we sort of simplify all these mobile elements into plasmids because it's easy to think about, but there's lots of different elements that move within each other even. So I think the likelihood, evolution would win still. You'd block one mechanism and you'd hit, and it would still move. It would just move through a different mechanism. But there certainly are efforts underway that I'm aware of. Yeah.
Thanks.
Great. So I think one of the purposes behind, or the principal purpose behind this symposium, was really to bring scientists and clinicians together in the room to really exchange ideas, to generate ideas. And so we have an opportunity over lunch for you to really network and talk to people and think about what your priorities are and whether anything this morning that's been mentioned stimulates an idea or an interest in a collaboration. So we'd really encourage everybody to do that over lunch. I really want to thank the speakers for some really interesting talks today. It's a great privilege, I think, to be in Melbourne and have this opportunity to mix with such diverse talent and expertise. I wanted to add, Ben, that you can strike another drug off your list because Nabriva's gone bust as well with lefamulin. So the only two drug companies I've been involved with was azithromycin and lefamulin. I've got lefamulin stacked in our pharmacy and got the email out like New Year's Day that the company's gone under. So we'll see what happens. Hopefully they'll get bought out by someone else. So, yeah, if you can join me in thanking today's speakers and network over lunch.
So we look.
Yes.